A very good afternoon to everyone. Before we begin with this webinar, I would like to pass on a few instructions for the participants. Please keep your audio on mute mode as well as your video switched off. Drop your queries for the resource person in the chat box for the smooth functioning of the session. Feedback link will be provided at the end of the session on your registered email address. E-certificates will be issued only after attending the entire session as well as after the successful submission of feedback form. With this, let me begin the program formally. Honorable resource person of this webinar, Dr. Harish Pad, honorable members of the management in absentia, respected principal Sir Dr. Vrajesh Patel, respected vice principal and IQAC coordinator Mr. Virendra Joshi, and dear participants. On behalf of Shet Piti Mahila College of Arts and Home Science, managed by Vanita Vishram Surat, and the IQAC of our college, I extend a warm welcome to all of you in this webinar entitled COVID-19 Infection, Immunity and Vaccines. Needless to say, how terribly COVID-19 has affected the entire humanity across the globe. It would be an understatement to say how desperate people and governments across the world are to get a vaccine for this pandemic. With introduction to vaccine and vaccination, this webinar will discuss the coronaviral behavior and disease progression in asymptomatic versus symptomatic patients. Based on the two recent research papers, the webinar will also discuss about the immune response to COVID-19. The lesson learned can be useful in developing vaccine strategy in future. So without much ado, let me request Ms. Krina and Ms. Bhumi to proceed with virtual lamp lighting. Thank you very much, Krina and Bhumi. Now I humbly request Dr. Vrajesh Patel, the principal of our college, to deliver the welcome address.
Very good evening to all. I welcome all the participants in this national level webinar on COVID-19 infection, immunity and vaccines, organized by IQAC and St. Mahila College of Arts and Home Science. I heartily welcome invite speaker Dr. Harish Pad sir, former Vice Chancellors of Sardar Patel University, Vallabh Vidyanagar. On behalf of our chairman, Dr. Pradeep sir, Vice Chairman Ashwin Mehta sir, Honorary Secretary Harish Mehta sir, Secretary Planning and Development Dr. S.K. Mohanty sir, and entire Vanita Bisram Parivar, welcome once again in this webinar. I congratulate academic coordinators Dr. Imran Surti sir and IQUC coordinator and our college vice principal Virendra Joshi sir organize this event. Wish you very happy learning to all. Thank you.
Thanks a lot, sir. Thank you very much for your words of encouragement and appreciation. It's time now for us to introduce the resource person of this webinar, Dr. Harish Pad. Dr. Harish Pad obtained his PhD in biochemistry from University of Delhi in 1978. From 1978 to 1995, he developed his academic career in USA. He was assistant director and associate professor at Northwestern University for biotechnology. In 1996, he returned to India and joined as a professor of biochemistry at MS University of Baroda. And in 1999, he joined as a director of B.V. Patel Pharmaceutical Education and Research Development Center in Ahmedabad. In 2007, he also took responsibility as the project director of NIPER, a Government of India's institution at Ahmedabad. He joined Sardar Patel University, Vallabh Vidyanagar as Vice Chancellor in July 2010 and continued as VC for the second term, that is to say, till it ended on July 31st, 2016. His area of research and specialization is cell and molecular biology as applied to health and pharmaceutical sciences. Dr. Pad has more than 170 research publications in reputed journals. He has more than 4,900 citations. Cumulative impact factor is 280, Edge index is 36, and I-10 index is 79. He had organized and attended many scientific symposia, meetings in India and abroad. He is a member of several editorial boards, societies, and IBS committees of pharma companies. Dr. Pad is the advisor to Government of Gujarat on the matters related to pharmaceuticals and biotechnology. He is also a recipient of B School Excellence Award and the Name in Science Award given by European Science Congress from Oxford, UK. He is a member of the Governing Council of Gujarat Forensic Science University, Gandhinagar, Central University of Gujarat, Gandhinagar, Association of Indian Universities, New Delhi, member of Court as well as Governing Council of Indian Institute of Science, Bangalore. Sir, we are truly honored to have you as a resource person for this webinar. Now I request Dr. Harish Pad to deliver the keynote address. Over to you, sir.
Thank you very much, Imran Bhai. Honorable Principal Sri, fellow teachers, and my dear students. Imran Bhai, I would like someone to enable the sharing of my screen so that I can show them. It says
Okay,
that
sir.
I cannot share it. Okay.
Yeah.
Okay. Okay, are you able to see this?
Yes, sir.
Is it okay? Then I'll proceed.
Yes, it's clearly visible. Yes, sir.
Okay.
Please do.
You know, it is really very nice to be with Vanita Vishram. This time virtually through internet. I have been there a couple of times, and it's really a pleasure to be with you all this evening. Now, the title of this webinar is COVID-19 Infection, Immunity and Vaccines. But before we talk about COVID-19, And since there are many students and I understand there are mixed population, the audience is with mixed background. So I'll try to be as simple as possible, although this is very technically very complex subject. And those of you who would have questions, please forward it to the coordinator so that I'll try to address them at the end. I think before we talk about COVID-19 vaccines, I think I would like to discuss some general background about what are vaccines, brief history. I'm not going to go very deep into it, but very brief history. From the perspective of India, where do we stand in terms of vaccinating our children, in terms of vaccine production? Where do we stand as a India? And then we will start with the discussing of Corona and COVID-19 vaccines. Now, all of us must have experienced that when we were kids, when we were very young, we all received some vaccines, polio, smallpox, etc. But that many of us may not know that the vaccines were developed in the early part of 20th century. And every country was using their own programs. Whatever vaccines were available, they were trying to vaccinate their own children through whatever vaccines were available. It was in 1974 that WHO introduced a program called Expanded Program of Immunization. And you can just see a simple fact. The proportion of the world's children who received vaccines earlier, पहले सिर्फ 15% बच्चे को वैक्सीन मिलती थी, इस प्रोग्राम के हिसाब से फिर 90% बच्चों को वैक्सीन मिलने लगी. So you can see the impact of WHO's program of EPI, or subsequently it was replaced by Universal Immunization Program called UIP. And India wanted to start it in 1987 and complete it in 1990. And this UIP program, not only smallpox and polio, but many other vaccines were added to that list of childhood vaccination. Unfortunately, we in India, we are not able to cover 100% of our children for all the immunization that is mandated through WHO's UIP program. Our coverage is about two-thirds, about 65 to 70%. We are still leaving out a good number of children. And individual coverage, individual means for particular vaccines, some coverage may be 8 to 85, but overall, all the vaccines are not reaching more than two-thirds of our children. Now, UIP program, Universal Immunization Program, has many vaccines that has to be included. And here is the list. You can see BCG, DPT, which is actually a mixture of 3 vaccines, diphtheria, pertussis, and tetanus. Oral polio vaccine, measles, hepatitis B, tetanus, etc. And even the last one, which is called pentavalent vaccine, which has a combination of 5 different vaccines. So the point I want to make it is that as the newer vaccines were available, the WHO tried to cover all the possible vaccine programs and tried to cover as much as possible to the world's children. Now, you can see that some of these vaccines were added very recently. In 2017, pneumococcal vaccine was added. Papilloma only in some areas. In Punjab, it was added in 2017, and other states are planning to add this vaccine. The point I'm trying to make is that this UIP program is very dynamic, and newer vaccines, as they are available and as they are needed, are included in this. But the challenge is to reach out to more children in India. I'll make that point subsequently. Many people question whether vaccines are really good and they are effective and they really save us from diseases. There are organized groups which oppose vaccinations, and they are in India, they are in USA, there are many other European countries also. There are people who doubt the effectiveness of vaccines. So this slide, जैसे आप देख सकते हो कि स्मॉल पॉक्स completely gone, polio completely out. Similarly, tetanus almost completely out, and diphtheria, pertussis, etc. So earlier we used to have 23,000 cases of smallpox every year, and now we have 0. And likewise, most of the diseases are controlled to the extent of 85 to 100% efficacy. So vaccines are really useful, and we should make it available to every children that requires it. Just one simple statistics, that infant mortality, that means the children who die before the age of reaching 5 years, under the age of 5 years. So you can see that in India we had 3.3 million, that is 33 लाख बच्चे इन्फेंट मोर्टालिटी से मरते थे. वैक्सीनेशन शुरू होने से अभी भी हमारे 13 लाख बच्चे की मृत्यु होती है, लेकिन आप देख रहे हो कि वैक्सीनेशन से काफी बच्चों को हम जिंदगी दे पा रहे हैं. और हमारी चैलेंज ये है कि ये सब वैक्सीन हर साल हमको करीब करीब 3 प्रेग्नेंट वुमेन तक पहुंचानी है, और 2.7 करोड़ न्यूबॉर्न, 3 crores of pregnant women and 2.67 crore of newborns. इन सब के पास हमारे वैक्सीनेशन पहुंचनी चाहिए हर साल. Unfortunately, we are trying to do very good, but we're still falling short of good vaccination. Yes, this slide shows that India has 1/5 of the children who die because of infant mortality. It's a good number of children who die because of infant mortality, approximately 13 lakh. 1.3 million children are in India who die because of infant mortality. And other countries, and we are the largest, the other countries are Nigeria, Pakistan, Congo, etc. And you can see many other smaller countries as well. So we have a huge burden of children for which the vaccine is available, but we are not able to make it available to our children. And we lose these children at a very young age, and which can be possibly saved. आप इस स्लाइड में देखो कि टोटली जो भी बच्चे हैं, nearly 1/3 of all unimmunized children live in India. कि हर में से एक बच्चा जिसको वैक्सीन नहीं मिल पा रही है वो इंडियन है. And that's a big challenge. And as I said, other countries are Nigeria, Pakistan, Ethiopia, Congo, etc. So we have problems of available vaccines, which we are not able to reach to the children who can benefit from this vaccine. If you see worldwide immunization coverage, this is slightly old slide, about 90% for some of the vaccinations. Hepatitis B almost 90%, and these all are measles, polio, etc. 90%. And some of the newly added vaccines, the coverage is less, but it is increasing. So point I want to make is that vaccines are effective. Second point I want to make is that we have vaccines that can benefit the young children, newborn children in India. But our reach to those children is very less. About 2/3, 1/3 of children not benefiting from the technology which is already available. Now, it's very interesting to see the early history in Indian perspective. I think I didn't know, and I'm sure many of you would not know, that plague and cholera vaccines were developed in India in 1897. That's almost 125 years back. So these vaccines were developed actually in India. At that time, it was a British India. And what Britishers had done, because many of their people who were in India, they were dying of infectious diseases. So they had started almost 15 vaccine institutes at that time, 125 years back. And when the Britishers left, most of these institutes were not really functioning well. There are a few exceptions, like Hopkins Institute, you might have heard of in Bombay. And I think in Baroda also there is one vaccine institute. So what happened after independence, this kind of research program went into back burner. And what they tried to do is to convert them into a production. That, okay, these institutes the Britishers established were not really functioning after independence, or they were rather neglected. And so we converted them into a production factories. And that was, in some way, it is good because we made use of the technology and the facilities that was available. But subsequently, even they were not able to adapt, because that's the case with many government companies. You know, there are PSUs, public sector undertakings. That is true in drugs, that's true in antibiotics, and that these PSUs really don't adapt to the changing technology. They don't modernize their facilities, and they fall into irrelevance period. And that's what has happened in India as well. And that is why the private sector players were allowed, and now they have taken Indian scenario of producing vaccines to a much higher level. And you will be surprised that now India is producing most of the vaccines which is procured by WHO. तो WHO और यूनिसेफ जो भी वैक्सीन्स लेते हैं उसमें से काफ़ी सारी about 1/3 is coming from India. तो एक तरफ से हम अच्छे प्रोड्यूसर साबित हुए हैं, that we can produce the vaccines for the established technologies. And we are also giving it to the other children through WHO and UNICEF. But we are not able to reach our own children. A good portion, 1/3 of our own children, we are not able to reach there. So you can see the contradictions that this line is a procurement from developing countries by WHO, and that by UNICEF, and that is by and large India. This graph, blue. And this is the contribution of developed countries. USA, Europe, Japan, etc. So their contribution has gone down in last 20 years, and contribution from developing countries, mostly India, has gone from 0 to almost 35%. So in last 20 years or so, we have become a good manufacturing source of the vaccines, which are fun and which are a part of WHO's program. You can see that every year our contribution to UNICEF's vaccine procurement increases almost 25%. So that's a very good ability that India has developed. And these are all the private players, Bharat Biotech, Biologicals, and almost 14, 15 of them. There are some PSUs, public sector undertaking, as I mentioned earlier. And there are some government-owned vaccine manufacturing units, as you can see, Pasteur Institute of India in Kunur. So these vaccines are, these companies are contributing to this growth of providing proven vaccines to UNICEF and WHO. So this is the kind of paradox that I wanted to emphasize, and I'm again putting it in front of you, that India leads in vaccine production, but not in vaccination. So we are not able to vaccinate all our qualified children, although we are very good in vaccine productions, and we are making money, but we are not, our children are not benefiting from the technology which is already available, and which is, so we have vaccines, but we are not, our coverage of vaccine to rural areas and poor people is still not. And you can see that if we have this vaccination, almost 60,000 children under 5 die every year in our country from vaccine preventable diseases. So here we have medicine, we have vaccines, and here we have children who need it, but we are not able to do that. So that's the paradox that Indians should keep in mind, and that it's good to be a good businessman, but also it's good to take care of the social sector and reach to the people who need it in India. This will become very interesting when we will discuss the corona vaccines. Now, whatever I have said so far is taken from this book chapter. This is the book, Vaccine in Developing Countries. And we have, myself and my colleague have contributed 2 chapters in this book. Whatever I discussed so far is taken from the first chapter in Vaccines in Developing Countries, the vaccine and vaccinations. It was published last year. And whatever I will discuss later on about what type of vaccine would be good for the corona, I think that concept is also described in one of the other chapters in this particular book. Now, I'm sure that you must be fed up with looking at this diagram of what coronavirus looks like. This is a little bit more detail that the virus from outside looks like this. And if you have a cross section, it looks like this. It has a single-stranded RNA as its nucleic acid inside the cover. Then it has a cover which is made up of lipid bilayer, but also has a number of proteins embedded in this. This particular protein called spike glycoprotein or S protein is very important for 2 things. One thing, it helps virus to enter the cell. And when it is able to enter the cell, it can multiply and then it can spread from that particular cell. If we block this particular protein, then we will be effectively blocking the entry of virus into our cell. And if we can block that entry, you will not have a disease. So all the efforts are to neutralize this molecule called S-glycoprotein, and neutralize either by chemically binding it so that it cannot bind to its receptors on the cell, or through antibodies, so that the antibodies, when it covers this area, then it will then prevent the entry into the cell of the host, that is our cells. And if we prevent entries, we will be able to prevent the disease. So all targets are aimed at this particular protein, this particular glycoprotein, so that we can minimize or inhibit the entry of the virus into the cells. I think it's the same protein. Our emphasis will be on this particular spike glycoprotein, which is a S protein, it's called. And the size of the virus is about 150 nanometer. So it's a very, very tiny virus. You can't even see with microscope. You need electron microscope to see this particular virus. Now, all this emphasis of this thing is on antibodies. This is the antibodies. This is a very general structure of antibodies against any antigen. Antigen is something that can bind to antibodies, and antibody and antigen reactions are very specific. So they can't, if it is unrelated compound, the antibody will not bind. But if it is a similar compound, the antibody will bind, and it will be, that binding would neutralize the effect of the antigen. This antigen is, as I told you, is this particular protein, S protein, that is the antigen for our context. So if we have antibodies and that can bind to that particular S protein, this complex will become ineffective. And when it becomes ineffective, it will not allow this virus to enter the cells, and you will be free from the infection from the viruses. Now, when we say that we want to make vaccines, what are the materials that we can use for making a vaccine? You can use any of this material to prepare a vaccines. They have some advantage, some disadvantage. Earlier days, it was the inactivated virus were used to make the vaccines, used as an antigen. It was inactivated, but it has a drawback that sometimes it can get activated and you might end up with the disease while you are trying to protect from that particular diseases. Or you have attenuated virus as a vaccines. You may have non-replicating, viral vector vaccines. In other words, they cannot replicate. So they are good as antigen, but they will not be able to multiply, and therefore it will not be, it cannot cause the disease, but it will cause the immunity. Similarly, most of the vaccines, they use protein subunit vaccines. So what it means is that the entire protein, in most of the cases, the entire protein is used to develop antibodies, and that it becomes the vaccine, which is mostly used. But the recent approach is that we need not use the entire protein as a vaccine. We can identify some portion of the molecule, and I will come to that later on. And those portions which are very good in antibody production, which has better immune response, if we can use them as a antigen, as a vaccine candidate, we will have a better immune response. We will have better antibodies which will protect the patient for a longer period of time. So in short, what I am trying to tell you is that there are number of approaches, strategic approaches for the vaccine production, and number of vaccine materials are also actually being used for COVID-19. As you can see that there are roughly 150 plus vaccines under the development for COVID-19, 150 plus. Although most of them are in exploratory phase. Exploratory phase means your ideas, and you try to convert ideas in and check it out, and then that is your exploration, that okay, it does work or it doesn't work, or it can be modified. And so that kind of exploration phase is, there are about a dozen candidates, or 12, 14 candidates, which are in preclinical stage. The preclinical means that we are doing the safety, whether it is safe in animal studies. So we do a lot of animal studies to see for two things: whether it is safe, and second thing, whether it is effective, efficacy. So whether it produces antibodies and protects the organism, and second, whether it is safe, whether it has any side reactions, etc., etc. So that's the preclinical. And then there are 3 stages of clinical study: phase 1, phase 2, and phase 3. So as you can see, there are a lot of explorations going on because it's a huge market, and there are a lot of competitions. There are political as well as commercial forces at play. So everybody is trying to make a vaccine. Although some candidates, about 12, 15, are advanced candidates and they have entered the possibility range. And that possibility range, we will see in the subsequent slide. Here, what I wanted to show you that whatever we discussed in the earlier slide, that you can use a live attenuated virus, inactivated virus, non-replicating DNA or RNA viral vectors, or you can use a replicating, you can use protein, recombinant protein, peptide base, etc., etc. So all of these possible candidates are being explored, and you can see that some of they are all over this particular range. So everything possible is being tried for development of vaccines for COVID. And the reason I told you that it's a very serious situation that the world is in, and we need a vaccine this way. I mean, we need a vaccine, and earlier the better. So all of those attempts are being targeted. We have, as I said, in preclinical stage, that is exploration and preclinical animal studies, more than 130 candidates. There are about 13 candidates in phase 1, phase 2, phase 3, and approved vaccine 1. That is from China, and that is only approved for using in military personals. Now, you might realize that this is a very dynamic situation. Every day we hear that 3 more candidates have entered phase 2, and likewise. So there is a lot of competition. This chart is driven by a lot of commerce and political interest. And of course, we have to handle the pandemic. We are trying to do our best, but then there is a lot of race. And when there is a race, sometimes it is healthy, sometimes it is not. It will raise several ethical issues that we will discuss at the end. But this is the scenario, and it's very dynamic. It changes almost every day. Yesterday, I read that 3 more candidates have entered the phase 3 trial in US. So it's a very hot and very dynamically contested area, and every player who has something to do, they are doing in this particular business. The earlier information that I showed you is shown here in a very different way. Here are the color coded, and if you can see that this red is a preclinical, and there are 4 Chinese companies which are in forefront of the vaccine development. There are 2 USA companies, there are Germany company, United Kingdom, Australia, and India. In India, we have these 2, also Serum Institute have entered recently. So Zydus Cadila and Bharat Biotech, they are now in phase 2 trial. This slide is about a month old, and you can see that here it shows it's a preclinical, but now they have entered phase 2. So they are already here, Indian candidates. So it's a very rapidly fast moving field. And this is the one that is from China that has been approved for use in the military people. So there's only one which has been approved so far. But hopefully in the next couple of months, we should have a couple of good vaccines in the market. Now, which are the major players? You can see that North America, that is USA and Canada, are the largest. Then there are China, also very competitive, Europe, and Asia and Australia. So they all are trying, and some of them are companies, private industry. You can say most of them are private industry, some academic institute, some public sector, and the non-profit sector, NGOs. They are also into this. Now, this is, I have said that these are recent additions, but these are actually a month-old addition. The Bharat Biotech and Zydus were entering phase 1 and phase 2. Now they are into phase 2 and ready for phase 3. Australian companies, Pfizer, and likewise. So there are, and this is the Chinese company which has approved for military use vaccines. So it's a very dynamic, and one has to keep track of what is happening there. Now, when we are thinking about producing a vaccine for COVID, we have to keep two things in mind. See, the vaccine is not a permanent thing. Why I said this? Because sometimes the antigen or the pathogen, it changes very rapidly. So if you have a good vaccine today, but if your antigen takes a different avatar, it changes itself in such a way that it will not be binding to this particular vaccine that you have produced. So it becomes ineffective. There are examples, for example, influenza. Now, influenza is a virus which changes every few months. So if influenza virus vaccines that you have today, next year in the cold season, in the winter, when you have an influenza wave, this vaccine will not be effective. And that is why WHO every year defines what should be the vaccine for this year's influenza disease. So what I'm trying to say is that even if we have a very good vaccine today, but if virus will change its form, that vaccine is not likely to be very effective. So we have to be aware about that thing. And second thing is, if we are exposed to virus today, and then we make antibodies, that's the natural body's reactions. How long we will be able to, how long we will be making that antibodies against that particular virus that we are infected with today, depending upon how long the antibody lasts in our body, that will define protections. So there are some cases where antibody will last for lifelong, smallpox, polio, that we have antibodies, and then we never have to worry about second dose of that particular disease vaccines. But there are cases where antibodies are very short-lived, and I will show you that that is the case with COVID. In COVID, we have both the problems. One problem is the virus, because it's a retrovirus, it mutates itself very fast. And when it mutates, after a few months or year, this virus will be very different virus, and your antibody that you have today will not be effective. And second thing is, even if somebody is infected today and develops immunity, is protected from the next infection, but that protection is only short-lived because you don't make antibodies for a long period of time. And this is some of the data that will challenge the design of the vaccine. And that is what the point I would like to emphasize, that we, in case of COVID, we are going to face both these problems. Our antigen is also very smart. Our pathogen is very smart. It will change its avatar very quickly. And second, somehow our antibody production is not for too long. It disappears in 3-4 months. So maybe after a year or so, you will not have a protection from the infection that you have today. So we are going to face very serious challenges when we think about COVID-19 vaccines. Anyway, but that's the way the vaccines are designed. And many of you must have heard about plasma therapy. Plasma therapy is nothing but a ready-made vaccine, ready-made antibodies that we isolate from recent patient who have recovered. So if the patient has recovered recently, we isolate his blood from the plasma, where we have a ready-made antibodies which can be given to a potential who can benefit, people who are exposed to viruses, and that will protect the people who have been infected. I thought I'll just explain the terminology. Now, I was telling you that some pathogens change very rapidly. As you can see here, that some pathogens don't change, like diphtheria, polio, hepatitis B, hepatitis A, etc. So if the antigens don't change, your vaccine is good and it will be good for 30, 40 years, almost lifetime, and you don't have to worry. Just take one dose in your childhood and you are protected throughout the life. There are some antigens or some pathogens which change every 10 years, pneumococcus, meningococcus. So you have to worry about after 20 years or so, 15, 20 years, so you have to see if you are prone to these diseases, you may have to take a second dose of vaccines. Influenza, I told you that every year the antigen changes, the virus changes. So when it changes, then you have to take that dose. If you want to protect yourself next year, you have to take every year this vaccine. And HIV virus, it changes every day. When the virus changes every day, that's why we have not been able to produce a vaccine for HIV in spite of our efforts for last 25, 30 years. So depending upon where your pathogen is, and my guess is that COVID-19 is somewhere here, that it may change in a couple of months, few months, 6 months at most. And that is why we will face the challenge of finding a good vaccines, which is effective for a long period of time. And the other variability, as I told you, our own response, like immune response, that how long our immune response lasts. And I will show you the data that it doesn't last for too long. It lasts for 3, 4 months, 6 months at the most. And so both of these problems are going to be there when we talk about vaccines for COVID-19. So if you want to control the infection through coronavirus, you have two approaches. I think we may have some we may have some friends from chemistry department. They are very familiar with making a heterocyclic chemical drugs. And these drugs will inhibit the binding of S protein, our antigen, with the receptor protein. And these are the receptors, ACE2 receptors on our cells. And through those chemicals, if we can inhibit these interactions, we can have a drug which will inhibit the infection. And the second thing is to have antibodies. That is through vaccines. So if we have antibodies, it will do the same thing. It will bind to this part of the virus. And because of this binding, and this is the antibody, it will not be able to enter the cells and it will not be able to divide, multiply, and that's why you get protected. Now, I was telling you that the immune response in case of COVID does not last for too long. Here is one paper taken from Nature Medicine. As many of you know, that it's a very reputed journal. This study was from China, originally from Wuhan, where the infection started. That they have taken about 37 asymptomatic people and 37 patients. The asymptomatic individuals were quarantined, but they were positive by RT-PCR test. So they were positive, but they didn't have any symptoms and they were under quarantine. 37 patients were hospitalized and then they recovered. And then they followed these two groups of people for 2 months, for 8 weeks. And then they measured what was the immune response corresponding to, what was the immune response in the beginning and at the end. And I'll tell you how that thing is done. And their conclusion is that immune response corresponds to the viral load exposure. What it means is that people who are asymptomatic had a very low viral load. The number of viruses reaching those people were relatively low. So they made antibodies, but they didn't make as much as the people who got a very high load of viruses and they got a better immune response compared to this group. So it says that your immune response corresponds to the viral exposure that you might have. And second thing it says that immune response is very short-lived. So they saw that in 8 weeks, that's 2 months, most of the antibodies that were produced in these two group of people was very low compared to initial. So they did this comparison. It's a very systematic study from China. And I will just briefly show you the decline of level of antibodies. There are two types of antibodies, IgG and IgM. And as you can see that both of these antibodies over a period of 2 months were down. We'll come to that next. But both of these antibodies were high in patients compared to asymptomatic people. This blue is asymptomatic people, 39 of them, and this pink is a symptomatic patients. So this is the level of antibodies in these people, and this is the level of antibodies in these people. And that is true for IgG as well as IgM. These are the two different antibody molecules, and they have a different role. What happens in 2 months, 8 weeks, that asymptomatic people, the level of antibody IgG is almost zero. As you can see here, this is the mean. The black line is the mean of antibody level. This is the antibody level. And the patients have some, but compared to this, they also lost most of the antibodies over a period of 2 months. So this is showing us that the production of antibodies or your immune response is very low if you are asymptomatic, and that disappears by the time of a couple of months. And if you are a patient, you will have immune response which is better than asymptomatic, but that all goes down relatively. So how long it will last, whether it will be zero after 6 months, we don't know yet. We have to see the long-term studies, but it is significantly down in 2 months' time. And these are the individual data points. As you can see that many of them are very close to zero in both the groups. Although this group has a better immune response, and some antibodies are left after the period of 2 months. This is actually a neutralizing test that, okay, you may have antibodies, but are they effective in neutralizing the antigen or virus? And they say that, yes, they are effective, but they are also going down over a period of 2 months in both this group people. So we can conclude that your immune response is there, but it's not going to last longer, and that your immune response is proportional to your virus exposure. This is a piece of additional data. I think I can skip that. So we concluded that your immunity will be there, but it will depend on how much viral load you have been exposed to, and second, it is going to go down over a period of few months. This study is also taken from a Nature, about a month-old journal, and this was from Columbia University in USA, in New York area. And what they did was they actually isolated antibodies from patients who had recovered, and almost 150 convalescent people over a period of 39 days. And they measured the antibodies in the beginning and at the end. And they did it slightly differently. They actually sequenced the entire antibodies. And what they found was that one, what was told by Chinese study, that yes, antibody level goes down dramatically. But interesting thing they found was that antibodies of a specific area of antigen, we call epitopes, they found in everyone, and every one of these 149 people, and they found that this binding of this antibodies to that particular site is very strong. Its KD is less than 1 nanogram per ml. So that particular insightful study that actually, uh, see, this is the entire protein molecule, that glycoprotein molecule. Now, when we are immunized, we can develop antibodies here, here, here against all of this domain, what we call epitopes. But what they found was that these three particular epitopes, or actually 4 or 5 of them, are very important because most of the people had antibodies against these epitopes. And these epitopes have a very tight binding. These antibodies to this epitope had a very tight binding. So that gives us a hope that perhaps if we have a vaccine which is using such epitopes, either these or similar epitopes, and if we have a vaccines produced from that particular part of the antigen, not the entire antigen, but the part of the antigen, or a mixture of them, what we call epimix. So if we have antibodies of epimix type, we can perhaps have effective vaccines for COVID. Now that's the last word in this particular area of designing vaccines for COVID. What it says is that we are facing two important problems. One is that antibodies do not last for too long. And second problem we have not addressed yet, that the virus also changes. I think that issue not come up yet because it's a very short 6 months experience with this virus, but that will come. But it also says that we should really think about adopting a non-traditional approach in vaccine design. If we want to design vaccines, we should have non-traditional approach of not using the entire protein or entire virus for vaccination, but we should use only those portions as identified by this study and a similar one. I think it needs to be elaborated and further explored, and a mixture of these epitopes perhaps can give us a better vaccine protection for this particular diseases. So most covalent plasma, this is the second study, obtained from individuals who recover from COVID-19 do not contain high levels of neutralizing antibodies. Now that has been demonstrated in several studies. So we have to live with this fact that our antibody will not protect us for too long. Nevertheless, rare but recurring RBD, that is a receptor binding domain, specific antibodies with potent antiviral activity were found in all the individuals that they have tested, suggesting that a vaccine designed to elicit such antibodies could be broadly effective. So in other words, we have to be innovative. We have to think about different approaches, not go by traditional approaches, but also try to incorporate this kind of understanding of how the immunity works in case of COVID. Our experience is very short, but we have to maximize the output from that experience, that how to have a better design for the vaccines and give the humanity a hope that, okay, pandemic has troubled us for 6 months now, but we are preparing our arsenals that we hopefully will be available with us in the next 6 months or so. And hopefully all of us will be immunized, although we don't know for how long it will help us. But at least if we are immunized once, and then hopefully virus either changes its mind and becomes mutated and becoming less infectious, less infective, And hopefully this pandemic of COVID-19 may become a history, but it's not in the near future that we should look for it. We should not be complacent. And we have to see that it will take a while before we really are prepared to handle this kind of pathogens. And the nature is also smart enough that once we prepare for this particular one, nature will invent something else to keep us on our toes for time to come. Since this is an academic group, I want to bring to your notice these two agencies which help vaccine development and application of vaccine. Gavi, which is a global alliance of vaccinations international. This alliance is made up of a number of agencies of WHO, UNICEF, WHO, many countries, many donors, including Bill and Melinda Gates Foundation. And their objective is that, what we discussed earlier, that India's coverage is only about 70%. So their priority is that our coverage, not only in India, but globally, should reach 100%. So every child who needs all these approved, WHO approved vaccines, every child should get that coverage and save some infant mortality and early death, and spare the lives of those children that we are currently not vaccinating. So their priority is not finding a new vaccines, but their priority is to increase the coverage. Now, for example, Sri Lanka has 100% coverage, and they are protecting their children, all of them, while we are their neighbor. They also have many difficulties. It's a rural area, jungle area. There are unrest in the populations, but still their coverage they claim is 100%, and we are not able to claim more than 70% coverage. So our priority is to increase the coverage. And the second agency is a relatively new one called CEPI, Coalition for Epidemic Preparedness Innovations. And their role is to find newer vaccines and be prepared for the next pandemic. And their role is to support the development of vaccines for COVID and all other unmet health needs globally. So we have two agencies. One helps the development of newer vaccines. The other helps the reaching out to people that we are not able to reach for the approved vaccination program. So we hope that, so what I would like to conclude here is that India is very good in vaccine production. As I have shown you, that we provide vaccines to the world, many vaccines to the world population through WHO and UNICEF. But we are not good at vaccination. We are not where we should be of reaching 100% vaccination of all our children. And it's a very painful fact to realize that, we have things, but we are not able to make use of it for our own children. That's a very painful thing. And COVID-19, the natural immunity will be very low and it will be short-lived. Traditional vaccines are not promising, may not be promising for too long. So better vaccines through epitope-based design or epimix. And while we wait for vaccines, we should not hold our breath. We should do what we can do now. And our best option now is precautions and wear mask and do all the things that the government tells us. And we should be prepared for, we should do what we can do. And that what we can do is take precautions. So I really thank Vanitha Vishram and the college for this opportunity to present this seminar to you. And final slide is that as you can see here, that we have to be in a stream of knowledge development as well as knowledge distribution. That's our responsibility as well as necessity for survival in the 21st century. So I think I'll stop here. I'll hand it over to the coordinator, and I'll be happy to answer any questions that you might have.
Thank you. Thank you very much, sir. Thank you for such a wonderful session. I read it somewhere that simplification of a topic for the general intelligibility is a great blessing, and today I witnessed the same. So you, for even a non-scientific person like me, it's a matter of great interest to understand such a topic which has created a lot of curiosity across the globe. So there are two questions from participants. One is from Mr. Sangadeep. He has a question: is ACE receptor same as ACE inhibitor?
No, no. ACE2 receptor is the receptor through which the virus enters the cell. ACE inhibitor is the chemical compound which will inhibit this process. Actually, ACE is a converting enzyme, and angiotensin converting enzyme. So inhibitor is the one which will inhibit this conversion, this protein angiotensin conversion. So inhibitor is something which inhibits that particular reaction of that particular enzyme. The receptor is the one that will bind to S protein, and it will internalize the virus into the cell.
I hope the answer of the query of Mr. Sangadeep is answered. There is one more question from Dr. Vinita Mode from our own college. Doctors have been using the BCG vaccine for protection against COVID-19. How effective is that vaccine?
I hope doctors are not using BCG vaccines, but in earlier studies, the observation was that people who were getting this infection, COVID-19 infection, were from Europe. There's not much in China, but China also had a good number initially. Then Europe and USA where most of the infections were found. And then India, a country like India, we were not having that many infections. So people thought that, why is this thing happening? At that time, the idea was that maybe BCG vaccines that Indians are immunized with is protecting Indian populations and that's why India didn't have a good number in the beginning.
That's true.
But now we know that that's not true. South America also uses BCG vaccines. China has stopped BCG vaccines, and people were explaining that that is why they had initially so many cases. But I think the reality is that BCG may protect to some extent that we cannot rule out at the moment.
Right.
But to count on BCG as a vaccine for COVID-19 would be a misjudgment on any doctor's part if they are doing that. And I mean, maybe it has not been established, very solidly. It has not been established enough to recommend BCG vaccines for general population.
Right. So there is one more question, from participant named Priyanka Soni. She asks, WHO, who has recently advised for the use of corticosteroids for the treatment of severely affected COVID patient? Why is it so?
Well, the corticosteroids are used to control inflammation. And what happens in COVID-19 is that there is a lot of inflammation in lungs, particularly.
Lungs. Yeah.
There is also a lot of bleeding. Actually, some anti-clotting and some clotting drugs are also being tried in severe patients, because there is a lot of micro bleeding, as we call it. So capillaries, they ooze out blood because of the infection. So it is the inflammation which the steroids will control. But that's not the solution. I mean, that may help to alleviate some symptoms for some time, but that's not the solution. And steroids have also their own set of side problems. So it may help in inflammation related. The most severe cases might be helped by such approach, but that's not the treatment for, that's not the prescribed treatment for COVID-19.
COVID-19. Right,
In
sir.
fact, we don't have prescribed.
Right, sir. I believe that, questions have, I mean, there are no more questions left.
I thought that some of you will ask me, when will I get vaccine?
Right, sir.
I'll tell you, it's going to be a very huge, dirty war when the vaccines are actually available.
Right, sir.
Do you know that America has already placed order for 3 vaccine doses for every citizen?
Oh.
American population is about 300 million people, and they have already placed order for 1,000 million. That's 1 billion doses have already been paid for by USA. Now, you will ask when it will come to India or when it will come to me. I'm telling you that is going to be a war, actual war over this one. Yesterday's news is that WHO has convened a meeting of these 200 some countries, all of these countries, to prioritize who should get vaccines first. And their thought is that we should give it to the first responders and medical staff, and then to aged people who are more vulnerable, etc., etc. So that's the logic. So they called a meeting that some of the developed countries, including US, they are not interested. They are not participating in it. They said, our money power will buy our vaccines and we don't need your prioritization. We'll buy it for every citizen. So there's going to be a really, a messy part of human nature will come out when we have a vaccine. And that's a very painful realization. When we will get, I don't know, I think our 2 vaccine candidates are likely to be out by December or so. That is, I'm talking about Zydus and Bharat Biotech. These 2 vaccines might be approved, let's say, by December. Now, that's assuming that they will prove themselves in the clinical trial in terms of effectiveness and in terms of safety.
Yes, sir.
If it happens, that will be somewhere in next year. It may be available. Now they can't produce 130 crore doses that they needed. So it will take another year so that there is enough for every citizen. So there is going to be a test of the will, that how we as India will decide to ration or to prioritize the vaccine distribution.
Right, sir.
I think it will test us. Some people will say that some political party members should get first and others should be behind.
Yeah.
And some community should get first, or some states should get first, and there are going to be, or there should be a quota that like our education, and there should be a quota for many
getting the vaccine.
So I think it will test us how good we are in controlling our our politics.
Right,
I
sir.
think it's going to test us.
Right.
And
Yes, sir. So
and
there
my
are few
real
more
question
questions.
is,
Yeah.
okay, I will come to that. My real
Yes, sir.
concern is that the effectiveness of vaccines is there.
Yeah,
Even
true.
if we have a good vaccines, it's not going to be, like smallpox that you take it once and forget about it. It's going to be every few months that you have to
Months.
worry about if the virus remains around us.
Right, sir. Right, sir. So the next question is
Yes.
asked by probably a student named Jinal. Uh, so will the dose of COVID vaccine be very heavy?
No,
It
it
is
should not
slightly
be
very
very heavy.
presumptive.
No, it will be a prick, so don't expect it will be oral or some other thing.
Right,
It will be injection,
right,
but it need not be a difficult injection. So that should not worry you. Uh, but it depends on what material, which vaccine is approved and which material they have used for vaccination. I think it will depend on that part.
on that. Okay. One more participant named Vrinda Ahuja has a question, sir. Many people are protesting against getting vaccinated, and they believe that their own immune system is capable of fighting this virus. So why is this vaccination being made compulsory? And what is, and is human body capable of fighting it without vaccination?
I think human body, as I showed you, that some people who recovered from infection, they had antibodies, and that
Antibodies.
antibodies will protect them. But for how long is the
How
question,
long?
and we don't know yet because our experience is very few months. So we have to see that how long the antibodies last, and whatever antibodies are left, whether they protect you from the virus. It
Right, sir.
remains to be seen how long the effectiveness of the natural immunity will be. And the vaccine immunity is also not likely to be very different in terms of a span of time that it will protect us. But we
Right,
have
sir.
to see. We
Yeah,
don't have a data whether any of this vaccine produces enough vaccine, enough antibodies to protect us for
to protect
year
us.
or longer, because we have
Right, sir.
not tested yet.
Right, sir. Probably the last question, sir,
And
of the day. Yeah.
no, in terms of Imran, in
Yes, sir.
terms of the last question,
Yeah.
people talk about herd immunity,
Yes, sir.
and the herd immunity means that okay, if we are, let's say in Surat, I don't know the population of Surat, but let us say it's about 40 lakhs, and
It's 55, sir.
if
55 present.
55 lakhs. No,
Yes,
it
sir.
is growing faster than I thought.
Fast. Yeah.
So if half of the people are infected in Surat, then the other half will be naturally protected, because when the virus is transmitted from a person to person, uh, I think the random chance is that wherever virus goes next host, that will be
Yes,
protected
sir.
if roughly half of it.
Protected.
Now that
Right, sir.
amounts to about 20 lakhs of people that have to be infected.
People
And if you
infected.
take the mortality rate of about 2%, तो 20 लाख का 2% आप गिन लो कितने हैं कि इतना mortality are you ready to wait for? And God knows how
That's
long
true,
that
sir.
that will be effective. So herd immunity is a good concept for other pathogens, other
Countries.
diseases,
Yeah.
but I think it's not like it. It's very elusive here, and we have to wait until we really learn a lot about it. But we can't
Okay, sir.
count on that. That's the point that I want to make.
Right, sir.
Yes, go on.
Yeah. One more question from a participant named Nirja. Uh, she wishes to know, the source of coronavirus.
Uh, I think a lot has been debated about where it
Debate.
originated. There are countries which are blaming each other. There are labs which are fighting over who did this, etc., etc. I think we don't know. We
Yeah,
don't know
exactly.
for sure where it came from, but its link is that wherever live animals are in contact with human, wild animals and live animals, when they are in contact with human, and particularly if people are eating live animals, that is likely to be the source of new viruses, because that has been proved in many other viral diseases earlier. SARS and a number of other viruses, where people are in touch with wild animals, and some of them are being used as their food source. That is expected to be the source of viruses, including coronavirus.
Right.
Now, I personally don't believe that somebody has engineered it and spread it. And that's my personal my personal thinking, and that it is not. It's unlikely that we can do that, because we have seen many viruses before this. When we didn't have capability to engineer viruses, we have seen that viruses do evolve, and they come and do. I don't know how many of you have heard about Spanish flu pandemic, which was
Yes,
about
sir.
100 years
1980.
back. About 100
Yes,
years
sir.
back, and crores of people died. अभी तो इसमें हम लाखों की बात कर रहे हैं, लेकिन उस जमाने में crores of people died, and you can't say that it was even in the lab. So looking at the history, I think our close contact with live wild animal is a likely source. Now, whether it's in China, USA, we don't know. But at the same time, I don't want to undermine the biological. I don't want to undermine the biological warfare. And some countries are really very bad at producing germs, producing toxins from the biological sources, and I think that's also something which it should worry us.
Yes, sir. So let us take up probably the last question of the day. Are there chances of getting reinfected? And if yes, how much? The question is from Tarvinder Jeet Kaur. Over to you, sir.
I think, as I discussed, that the natural immunity, those people who are infected would have antibodies, and that antibodies are likely to protect them for a few months. We don't know exactly how long. But now we are seeing, now that we have about 4 or 5 months that people have recovered from the infection. Now we are hearing every day that some of those people are reinfected.
Okay.
So if you extrapolate this work that I described in this presentation, that our antibodies would protect us, whether it's through natural immunity or through vaccine. Our antibodies will protect us for a few months. We don't know exactly how many, but 6, maybe 8, maybe 1 year, etc. That remains to be seen. Right. But then, yes, once your antibody is out, is gone, you are prone to reinfection.
Right, sir.
That's why we have to learn a lot about virus. Our experience is very brief, relatively, and I think it will take many couple of years to really compile the data and learn from how we should handle this situation. But until then, precaution is the only thing that we should.
So let us end the discussion with one last question, from one of our own student. What do you think we should do to create a global society that will be less vulnerable for future pandemics to occur? The question from Sherin Jose.
I think, Sarin, I'm not worried at the moment about future pandemics. My worry is now. My worry is that why many people are not wearing masks, why many people are not taking precautions that you can do now. Once we have a human behavior under control, not control, a kind of military kind of control, but a self-discipline role, I think we can handle many things. And whether it's any pandemic or whether we have any other, any kind of difficulty, But the key thing in pandemic is the human behavior. Knowing so much, we have been told so many times that we don't have any drugs, we don't have any vaccines, and even if vaccines come, that's not going to be a really magic. Until then, we have to use the precautions that we can use. I've seen people that about 10% of people in the marketplace are wearing the mask properly.
Right,
Other
sir.
40% have it hanging somewhere here, not protecting, and other 50% are not even wearing masks. Now,
Really?
this is a risky behavior. Maybe some people, I think there is no excuse. I was thinking that maybe some have to go out every day, but not wearing mask is not really an option. But many, what we are seeing in India is also seen everywhere. In USA also, I think there are organized protests against wearing masks. That it's a my personal liberty and my human rights, and who can tell to wear a mask, etc., etc. Even the president, he has he's worn mask only twice, and that is for PR re-election campaign. Not really seriously, he has not been advocating use of mask. So I think we have a major problem in human nature, in our social behavior, in our cooperations. You see, when pandemic was at its peak, these two major countries were fighting at each other that, okay, you brought the virus, and they say, no, your military people brought the virus. And you see, that exposes the dirtiest nature of mankind. And when we have this, how can we expect that, let us be prepared for the next pandemic? Even we not
Next one.
behaved, and we are not even behaving now.
Now,
You
right,
tell
sir.
me, you tell me why this country, cities like Surat and now smaller cities like Rajkot are now hotspots.
Right,
Can we
sir.
not take experience from Ahmedabad and Delhi?
Delhi. Right, sir. That's true.
I don't know if I answered him, but that's my response.
Okay. Uh, okay. There's still a few questions. I can take one or two more. There is
I don't mind. I'm available. It's up to
fine,
you.
sir. There is a dietitian, Karishma Patel, who has a question. After getting recovered with it, should we still taking care about our diet?
Oh, I think diet has nothing to do with only corona. Diet is everything to do with your life, every aspect of your life. Why should you abandon a good, healthy diet practice because corona is out?
Right, sir.
Uh, it prepares your body and mind and many things. So there is no reason to link corona with a good diet.
Okay. Sir, I think on this note, we can wind up our discussion. I now request Mr. Virendra Joshi to propose the vote of thanks.
Good evening, everybody. On behalf of
Good evening,
CTC
sir.
Mahila College of Arts and Home Science, managed by Vanita Visram, and the IQAC of our college, I extend my deepest sense of gratitude to today's resource person, Dr. Harish Bhatt, sir. Sir, your presentation has given us a clear insight based on the research findings in this very important topic and a highly controversial one also. It was an enriching experience overall. I also thank our ever supportive management as well as principal sir for supporting and encouraging us to come up with such engaging programs. I would also like to thank all the participants of this webinar for showing their interest and enthusiasm. I would like to thank all my colleagues and faculty members for being a part of this webinar. Last but not the least, I convey my gratefulness to Ms. Bhumi Desai, and Ms. Krina Desai for the extensive technical help. Thank you all.
I may add that if somebody wants a copy of my presentation, you can send me email. I had my email address on my presentation, and I'll be happy to share the presentation if somebody requires.
And sir, if there are a few more questions, which comes our way, I'll, forward them to you. For sure.
Okay.
Yes, sir.
I will try. If they are very difficult questions, you can answer them. If they're easy one, you forward it to me.
That's an understatement, sir. Thank you very much, sir. Thanks for being online. Thanks for taking up this work. Thank you very much, sir. Pleasure.
Okay.
Pleasure. Thank you all.